Hey everyone, so this is the week 5 lab. This is the only assignment in week 5, which tells you it will probably make you want to pull your hair out. Let's look at the rubrics because you know they go below on the labs. As always, you are get an S post for sharing your table. Looks like there's 2 in this one, and you have your sample paper and your rubric. So let me open up that sucker. Okay, so there's 2 exercises. Each exercise is worth 3 points. You're going to have 2 points for format. So let's do format first. Complete sentences is a point, grammar, all that stuff. Make it legible. If I have to work to read your stuff, I will be unhappy, basically. And then APA, yes, you need references. For the first assignment, the table is 0.3 points. Each question answer is 0.3. The second one, the table is 0.5, and each question answer is 0.5, which lets you know this one's probably shorter. Okay, so let's scroll down, fill in the distance. Do check that manual, man. It has pictures. It's so helpful. Figure out your rates, all that fun stuff. So what molecules did the plant produce when exposed to light? Make sure you remember that not just photosynthesis is going on. During photosynthesis, what resources are consumed, what part did the light play in the reaction, what happens when it's dark. So what's happening when it's dark also happens when it's light. It's just now only the dark thing happens in the dark. Okay, so what was produced, thinking about the experimental design, what variables are present, and how could that cause different results from student to student. I always like to think of just what you think could change. What could be the changes, right? We're not going to all have identical results, so why would there be changes? Then, how would it be varied if you use a different wattage of bulb? And then, how is chlorophyll involved? Here's where I would probably need a reference, personally. And then, starting position, finishing position, change in movement and time. And then, are we still exercise 2? Oh yeah, there's 8 on the other one. There's 5 on this one. I was like, really? Why are they worth so much more? Yeah. So you blew through the straw. What gas did you add? What color change did you observe? Then you're going to talk about the blue stuff and tell me what happens. And then how did the control do stuff? If your control did not change color, do you think it would have eventually changed color? So what's really happening in your control? If you're not sure, look at that lab manual again, and of course you can always Google. Describe the relationship between photosynthesis and respiration. This is where I definitely would probably be using a reference, if not just the lab manual, something else, okay? So there you go. It's going to make you want to pull your hair out a little bit just because it's a little bit more of an in-depth experiment than some of the other ones we have. Not so much on what you're actually doing because it does have pictures, but like understanding what it means. So hopefully this helped you and you understand what everything's allotted for point wise, and you guys will all do great. And if you're worried, as always, send me a draft. Okay, I hope it helps.